Hello, my name is Dr. Roderick L. Roll, and today I will be talking to you about the cytoskeleton. The cytoskeleton is a network of fibers extending throughout the cytoplasm. It organizes the cell's structures and activities. It also anchors many organelles. The cytoskeleton is composed of three types of molecular structures: actin filaments, microtubules, and intermediate filaments. The thickest of the three is the microtubules. The smallest of the three is the microfilaments. The microfilament is composed of actin molecules. The one that's in between the two is called the intermediate filament. This slide gives you an overview of the microtubules. This slide gives you an overview of the intermediate filament. And finally, this slide is the smallest, the microfilament. This is an overview of the microfilaments. We're going to go through each one of these and talk about them extensively. The first one we'll talk about is the microtubules. The microtubules are hollow rods composed of tubulin dimers. So if you look at the tubulin dimer, it is composed of an alpha tubulin and a beta tubulin. The function of the microtubules is to help give shape to the cell. We're looking at an animal cell, and in this animal cell you see these yellow wires that appear to run throughout the cell. These yellow wires are microtubules. It helps give shape to the cell. This cell appears to be round. It is able to keep its roundedness because of the microtubules. So besides giving shape, the microtubules also will guide movement of organelles. You can see that hollow microtubule, and attached to it, it appears that something is walking along the long hollow tube. The thing that is walking is attached to a vesicle, and the vesicle contains something in it. So the thing that appears to be walking is called a motor protein. So motor proteins like kinesin and dynein will move along the microtubule to produce this vesicle motility. This slide shows the microtubule made of alpha tubulin and beta tubulin that forms a hollow tube. You also see the vesicle being pulled from left to right along this hollow tube. Energy is required, and you have a motor protein that is pulling the vesicle along as it walks along this hollow tube. This slide is an electron microscope image of this phenomena occurring. The third thing that microtubules will do is it will help chromosomes to separate during cell division. The common name we will call the microtubules during this process is a spindle fiber. So the spindle fiber will attach to a replicated chromosome, and it will move it around within the cell. This is going to help the cell to divide. In many cells, microtubules grow out from a centrosome. The centrosome is nothing more than a microtubule organizing center, or you can say it produces spindle fibers. So the centrosome is going to replicate and move to opposite poles of the cell during mitosis. Then this microtubule-organizing center is going to produce microtubules that are going to eventually attach to the replicated chromosome. Once attached, it will move the chromosome. In animal cells, the centrosomes have a pair of centrioles. So one centrosome has two centrioles. So once the centrosome replicates, you should have 4 centrioles within an animal cell. The centriole has microtubules arranged in a ring of 9 triplets. This is a transmission electron microscope image of centrioles within a centrosome. Notice the 9 triplets. The fourth thing microtubules will do is they will help cells move, or they will help cells feed. So microtubules control the beating pattern of locomotion appendages called cilia and flagella. They differ in their beating pattern and their lengths. The flagella is typically long and the cilia is typically short, and they have a different beating pattern, but both of these are made of microtubules. This shows a flagella being used by sperm. It is a motile appendage that helps cells move. The cilia can line things such as the trachea, and it can move debris like bacteria and dust from out of the lungs up into the pharynx where you can cough it up. So how do those things move up the trachea? It's through this rhythmic movement of this appendage that is attached to certain cells that line the trachea. So both locomotion appendages have a core of microtubules covered by a plasma membrane, or better yet, it's called a sheath. They also have a basal body that will help anchor them into the cell membrane. Now we will look more closely at the detailed structure of the cilia and the flagella. Just like the centriole, which had a certain arrangement of microtubules, the basal body, the attachment of the cilia and the flagella, the basal body contains a specific arrangement of microtubules. You will see 9 triplets arranged in a circle with nothing in the middle. In reference to the shaft of the flagella or the cilia, that shaft contains 9 doublets in a circle with 1 doublet in the middle. The cilia and the flagella will have a motor protein called dynein. This dynein is going to drive the movement of the flagella and the cilia. It is going to help them to bend. In this image, we're looking at the dynein arms. It's going to help the bending of the cilia and the bending of the flagella. So how does the dynein move the flagella and the cilia? It is going to move it in a process called walking. So in an alternating fashion, the dynein arms are going to grab, move, and release the outer microtubules. This is going to cause a bending movement of the flagella and the cilia. This image shows it in better clarity. You see the doublet and you see the dynein arm that attaches to the opposite doublet. This dynein is going to move, therefore moving the doublet either up or down. This is going to create a bending motion for the flagella or cilia. Also involved in the rhythmic movement of the flagella and cilia is protein cross-links called nexin. This nexin is going to limit the sliding effect of the microtubule doublets. Forces exerted by dynein arms will cause a doublet to curve. This will therefore bend the cilia and it will bend the flagella. The second category of cytoskeletal proteins we will talk about is the intermediate filaments. The intermediate filaments support cell shape and it fix organelles in place. The intermediate filament is a more permanent cytoskeleton fixture than the other two classes. This filament consists of a protein from the keratin family. The intermediate filament makes up the nuclear lamina by forming a net of keratin that lines the inner surface of the nucleus. If you look in this image below, you can see the nuclear lamina. This was taken from a transmission electron microscope. It is nothing more than a net of keratin molecules. The intermediate filament also binds to the anchoring junction called desmosomes. By binding to the desmosome, it is going to help reduce internal tension. The desmosome holds adjacent cells together. Additionally, the intermediate filament is going to anchor the microvilli in place. So the microvilli has a structural cytoskeletal protein called microfilaments that make up its core. This core is attached to the intermediate filament, which holds it in place. Remember, the microvilli helps increase surface area in the small intestines so that more absorption can take place. In this image, we are looking at a transmission electron microscope image of the microvilli. Remember, we have the microfilament that makes up the core of this projectile that protrudes off of the small intestinal cells. What anchors it is the intermediate filaments. The third cytoskeletal protein we will talk about is the microfilaments. The microfilaments are solid rods of twisted actin subunits that are roughly 7 nanometers in diameter. They are well known for cell motility. Tissue motility or organismal motility is achieved by the sarcomere. So this image is a sarcomere that shows you the contractile unit of a muscle cell. It is composed of actin filaments and it is composed of myosin filaments. In this image, we're looking at multiple sarcomeres. So multiple sarcomeres make up a muscle cell. Again, we're looking at the contractile unit within the muscle cell, which is composed of actin filaments, which are the thin fibers, and the myosin filament, which is the thicker fiber. In muscle cells, thousands of actin filaments are arranged parallel to one another. These filaments slide across the thicker myosin filament. So in the image below, the purple filament is the actin filament, and the green thicker filament is the myosin filament. In order to get the muscle to contract, the Z disc in this image will get closer to one another. So that movement of the Z disc is called the sliding filament theory. The thicker myosin filament interacts with the thinner actin filament. This will contract the muscle. Therefore, this creates movement on the organismal level. Now, on the unicellular level, actin and myosin are involved in things such as amoeboid movement. When an amoeba moves, it will push out its cytoplasm into what is called a false foot, a pseudopodia extension. This is achieved through the interaction of the actin and myosin. Additionally, we will look at cytoplasmic streaming inside of elodia or elodia. So this cytoplasmic streaming is a circular flow of cytoplasm. This is also achieved by the interaction of the actin protein.